Hi everyone, so today we're going to be talking about DNA structure. The genetic material actually needs to exhibit four characteristics. It needs to be able to replicate, to store information, to express that information through RNA to protein, and also allow variation by mutation in order to evolve. So as we know, the central dogma of molecular genetics is DNA makes RNA makes protein. So we have DNA makes RNA, which is called transcription, and RNA makes protein, which is translation, and that is the central dogma of molecular biology. Now, this is the structure of DNA. As you can see on the outside, there is a sugar phosphate backbone. So we see sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate on both strands of the backbone, and the sugars and phosphates are held together by phosphodiester bonds. In the middle of DNA are the bases. So G always pairs with C and is held together by 3 hydrogen bonds, and A always pairs with T and is held together by 2 hydrogen bonds. Now, if we go deeper into the structure, we can see that the diameter of the DNA all the way across is 20 angstroms. One complete turn, which is 1 and complete, so it's 360 degrees, is 34 angstroms, and there are 10 base pairs per turn, so the distance between each of the adjacent base pairs is 3.4 angstroms. So if you consider the DNA as a ladder, then the rungs of the ladder are made by the bases, which are held together by hydrogen bonds, and the handrails are made of the backbone, which contains sugar and phosphate. So if you look at the center of the molecule, you can see that it's not perfectly symmetrical, right? So you can see that there's a major groove which has a depth of about 22 angstroms, and on the other side there is a minor groove which has 12 angstroms. And we also have to remember that structure dictates function. So the major groove basically binds transcription factors. Here we can see the structure of a nucleotide. So a nucleotide contains a phosphate, a pentose sugar, and a base. So this unit is called a nucleotide. And the G's and C's, as I said, are held together by hydrogen bonds, 3 hydrogen bonds, and the A's and T's are held together by 2. As you can see, the guanines and the adenines contain 2 rings, whereas the cytosine and thymine only contain single rings. You can see that more clearly here. We have adenine, which contains 2 rings, and we have thymine that contains the 1 ring. And here you can see that adenine and thymine, A and T, are held together by hydrogen bonds. So there are 2 kinds of nitrogen bases. Adenine and guanine are known as purines, and cytosine and thymine are known as pyrimidines. Uracil is present in RNA, and that is also a pyrimidine. So what's the difference between a pyrimidine and a purine? So the pyrimidines are single-ringed structures, as you can see here, and they are cytosine, uracil, and thymine, whereas purines are double-ring structures, and they are guanine and adenine. So DNA and RNA both contain adenine, cytosine, and guanine. DNA contains thymine, whereas RNA contains uracil. So as I said, nucleotides contain a nitrogen base, a pentose sugar or a 5-carbon sugar, and a phosphate group. And these are the building blocks of DNA. So DNA is a polymer. Each monomer contains a nitrogenous base, a pentose sugar, and a phosphate group, and is known as a nucleotide. So you have a phosphate group, a pentose sugar, and a nitrogen base, which is a nucleotide. So just the one phosphate, not three like shown here, one phosphate, one sugar, and one base is a nucleotide. A nucleoside contains the base and the sugar and lacks a phosphate, whereas a nucleotide contains the base, sugar, and the phosphate. So here you can see how the phosphate, sugar, and base are put together. So this is the pentose sugar. It's a pentose because it has 5 carbons. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon number 5. Okay, this is the base and this is the phosphate. And the 5' and the 3' ends are determined by the numbering of the carbons. So 1, 2, 3. So you have a phosphate here, 4, 5. On the 5th carbon, there is also another phosphate. So RNA contains ribose as its sugar, where DNA contains deoxyribose. So what's the difference? So ribose, which is present in RNA, at the 2nd carbon, it contains a hydroxyl group. But in DNA, we have 2-deoxyribose. What does that mean? Deoxy means without oxygen. So the 2nd carbon, so you number them like this, 1, 2, 3, 4, and 5. So the 2nd carbon does not have an oxygen. Therefore, it is deoxyribose. So the sugar in DNA is deoxyribose.